All right, 7.3 video 1. Let's go. So essential idea. The information is transferred from DNA to mRNA and then goes into a amino acid sequence back to our basic, used to be called the central dogma. We want to get away from dogma. Dogma is not backed up by evidence, but this is. So DNA again to our mRNA, making it to amino acid sequence. Remember, that is our protein. So it's all information. Info. This is more info. And this is more info. Okay? In terms of DNA and RNA, you're looking at nucleotides. Your A's, T's, C's, and G's, or of course with the U with the RNA. And amino acid and proteins are based off that amino acid sequence. Remember, there are 20 different types that humans use. I just learned that there's more. There's hundreds of amino acids, but living things only use 20. Mind-blowing. Why is that? So that's the essential idea that we're going into. And in terms of nature of science, computing, this is something that really blew my mind back in 2003. I think I took a bioinformatics workshop. It really had a profound influence on me. Using computers, using the digitized information that scientists are able to get from DNA to find genes, to understand in really fine detail the structure of genes, or of proteins in particular, and then based on just the gene itself, scientists can determine the shape of the protein that gene is coding for, and being able to research disease. A lot of it is still really ongoing, this research, and what's really cool, when you all get older, you're going to see the fruits of it. And one of the main fruits from this basic information from DNA and RNA is the development of these RNA vaccines that we've all been so dependent on to help to alleviate the amount of people that have been dying and suffering from COVID. And it's going to be like that in the future. Just totally awesome. Think about the common cold. I don't like it to get the cold, but this research that's been spurred on by COVID, maybe we'll be able to get a vaccine once or twice a year, so you don't have to get the cold. Think about that. And we'll be showing you, or I'll be showing you information later on in the video. I think by the video too. And this is show you how to use this. This is anybody can get this information. You can see these are the human chromosomes 1 through 22 autosomes, and then of course the X and the Y. This is basically your human genome. You can search. You'll see you can't see. I searched it for cystic fibrosis. That is a genetically transmitted hereditary fibrosis. And it'll show you where scientists have discovered genes that give you cystic fibrosis. And based on all this, look at all this information. I have a tiny little bit of idea of what all this means, but probably like 0.05%. But look at all these numbers and graphics. So cool. I wish I had. I need to make time to do that. Okay, but first, to make protein or make your amino acid sequence from DNA, first you have to have the ribosome come in on the mRNA to start building it. So the first thing, that messenger RNA, from the DNA binds to the small ribosomal subunit. and that's 30S typically. This first part is 50S. So add them together, you get 80S. That's why our ribosomes are called 80S. But the small little bit, it looks like a hot dog bun, binds to the messenger RNA. And then you'll see it always starts with methionine, M-E-T. This is called the anticodon. This is called the anticodon. So the codon is on the messenger RNA, and the anticodon is on. I was right. The codon. The anticodon is. Let me go blue. Maybe that was messing me up. Anticodon is on the tRNA, and the codon is on the messenger RNA. And once you have the this methionine, this initiator tRNA carrying the methionine, binding to your start codon, then you have that large ribosomal subunit arrive. And then look, you got the initiation complex is ready to go. And then what'll happen is another tRNA will bind to the A site. And then once it comes in, it'll be carrying another amino acid, and then a bond will form between these two amino acids. It's called a peptide bond. Eventually, you're just going to get this long chain of amino acids that are being built. They'll eventually become the polypeptide. We'll see that on the slide in just a moment. But first thing first, we have that initiation complex completed. Go to my next slide. There we go. And you see here that tRNA is coming in. This is the amino end of the polypeptide. Just this long chain. These are individual amino acids. You need some power from GTP to create these bonds. And here we go. This is that peptide bond that I mentioned. Peptide bond being created. And once that is created, you'll see it shifts from that previous tRNA to the new tRNA. Our ribosome will shift over, and this old tRNA will be popped out, and it'll be recharged, so to speak, with a new amino acid soon. Be recycled. You don't want to make a whole new tRNA, so it'll be recharged, and then it's ready for the next tRNA. Once it's popped out that old tRNA, it'll just keep doing the cycle over and over until it's finished building it. Now in our body, for us, this is happening out in the cytoplasm. Alright, that this RNA was produced inside the nucleus, leaves the nucleus, enters the cytoplasm, and our body is going to be producing these. Now when it's over, when the when that gene has been fully transcribed and it's all done, a stop codon will be arrived at. So it'll keep cruising, boom, then it sees a stop codon. There's 3 different kinds: UGA, UAA, and UAG. And that pops into that A site, this release factor. So there's no say stop amino acid. Like there is a start, so it is confusing. There is a start amino acid, but there is no stop amino acid. There's just this released factor that pops into that A site and then tells the complex to break apart. So the two subunits come off that messenger RNA, and then the polypeptide is released. That's what you see here. Polypeptide. It's all one word. I made it look like two. And then these will be recycled as well, these large and small subunits, and the messenger RNA will be reused. Well, the messenger RNA, a certain amount of times before, I can't remember how many times it's used before it needs to be recreated. Remember, you don't want your body, that's wasting, to have to recreate everything. So two types of ribosomes, bound and free. Free ribosomes float around in the cytoplasm, and they typically make things that will stay in the cell. Bound ribosomes are on the endoplasmic reticulum, and they'll typically make things that will leave the cell. So free, primarily within the cell, making stuff that stay inside. You kind of think of it like, making things. If you look at a business standpoint, businesses making things that will stay inside the United States. And then membrane bound, things that will be made and then shipped outside of the country. Like our agriculture is one of those things in California. Most of it stays in the United States, but some of it is shipped outside the country, like nuts, pistachios, almonds. Those are pretty lucrative, so a lot of that will be exported. But this is a pretty cool diagram showing you that. Here is the free ribosomes, proteins, making proteins. They'll go from the nucleus, the mitochondria maybe. Very important for all cells. And then for plant cells, you have chloroplasts. Ooh, let's do this. Bring in some color. Ooh, mitochondria, gotta fix that. And then peroxisomes. Those are small organelles that break things down. They have enzymes that break down old stuff, waste products, and they make peroxide as they do that. Now over here, the membrane bound. Now remember, these are typically on the ER. Remember the rough ER and the smooth ER. So the rough ER is considered rough because it's got mitochondria all over it. So there we go. Much better handwriting. Rough ER. And these are making proteins that'll say go to the plasma membrane. So it's kind of like in the cell, but outside the cell. It's on the outside membrane. Now it's kind of confusing when we talk about membranes. Plasma membrane is a membrane. We call it the plasma membrane because that's on the outside of the cell. That's surrounding the cell. You also have plenty of membranes inside the cell. Secretory vesicles. Those vesicles will be produced. It'll go into that secretory vesicle, say if it's insulin, and then be exported. So, e.g., insulin in your pancreas. Then endosomes, eventually turn into lysosomes, usually, which do stay in the cell. So, again, not everything is 100% cut and dry, all the same. So here we got the bound ribosomes, a little more information on that. produced in the bound ribosomes on the RER. R for export. Got a little diagram showing that. Ooh, ER, that's right here. Rough ER, all bumpy, right? And this is a secretory vesicle going through the Golgi, getting processed, and then that protein then goes outside of the cell, showing budding right there. Pretty cool. Or creating lysosomes, and they need enzymes, which is what we're being. Remember, enzymes are types of proteins. Type of globular. Well, that'll come up in a minute. Globular, pretty globs, like a glob. You know, compact shape, circular, I guess, spherical. You also might kind of call it. And proteins produced by free ribosomes are for use inside the cell. So, just the difference between bound and free ribosomes. And IB wants you to know that prokaryotes can do their translation, you know, faster because they have no nuclear membrane. So here we have a prokaryotic cell, no membrane. The DNA is just floating around inside of there. So you can have your transcription and translation right there together. Transcription, which for us occurs in the nucleus, and then translation. So all happening out in the cytoplasm. Boom, boom. And remember, if you see a picture like this on an exam, you should be able to label it. Okay, this big, like the hamburger, the top bun and the bottom bun. I should make hamburgers like that. That'd be a fun experiment. Making ribosome burgers, so a really big fat top bun and a little skinny bottom bun. And then you could make something, anyway, the meat or whatever you put on there could be the RNA. Oh man, and then what could be coming off representing the polypeptide? I don't know. But here we have the ribosome. And I'm getting hungry. It is dinner time. This is 4:46. Polypeptide is here. And the messenger RNA, mRNA, right there.